I just hit the go live button, and we are now live.
We're now live. Okay.
So you are on the air. Welcome
Okay.
to Evidence the Evolution Show featuring Ian Chen, and this week we also have Barry Desborough here to talk to us about some of his favorite evidence for evolution.
Mm-hmm.
Some that I know Ian really likes, the ERVs.
Yes. Yeah.
So tell us a little bit about yourself.
We've got an expert today, though, Austin. So we've got an expert. Don't forget that we've got Barry, who's an expert at this as well.
Yeah.
So, do you want me to start things off speaking about ERVs?
Yeah, that would be great, Barry.
Okay.
Tell us a little bit about yourself first.
Okay.
And then we'll talk ERVs.
Yeah, quite happy to. Oh, and by the way, when this comes out on a YouTube or something, I hope to be able to put some links to my blog and to material that people can refer to, because I'll be talking about these matters, and we can go in greater depth after.
Send those to me, and I will put them in the description for sure.
Okay. So, a very brief rundown. I started out my adult life training to be a school teacher, and when I was looking for a place to train to be a teacher, the place that came up was actually a Christian founded college of education. They were part of the British education system. The British education system, when it was nationalised, took on a lot of church-based schools and colleges and so forth. So this was in Canterbury in Kent, important religious centre for Britain. And all the staff basically were all Christians. They had no trouble whatsoever with evolution. They even took us to Darwin's Down House on a day trip. We discussed evolution there. It wasn't such a big deal. And it came to I changed career. I worked as a school teacher, then I became a software engineer, and I was fortunate enough to make enough money at that to retire early. I thought, what shall I do with my time? I'd like to study human nature, and I got into evolutionary psychology, and I was discussing this with mostly American academics. And that was about the age of 50. I'd learned that people, particularly in America, a lot of people do not like the idea of evolution, and I'd never heard of this. I thought it had all died out in the in the 19th century. I was astonished. So I've spent an awful lot of time discussing things with and arguing with creationists, and eventually I came across endogenous retroviruses, and I thought, God, this is absolutely the most powerful line of evidence you can possibly have in favor of the fact that we share common ancestry with other kinds of animals, of creatures. So I've made that my special study, and a large part of my blog is to do with explaining endogenous retroviruses and dealing with questions or objections that come up from the creationist side. So there we are. I've made myself a bit of a name as somebody who knows more than the average amount of stuff about surrounding.
We'll certainly put a link to your blog in the description too, because your blog is amazing. It's incredible.
Thank you.
You have so much content there, and you've really addressed an incredible amount of creationist arguments, and it's fascinating how many creationist arguments endogenous retroviruses just absolutely knock down and drag right out of the ring. It's amazing.
Yeah, I have a page with links to virtually all the articles that creationists have written concerning endogenous retroviruses, and I critique all of those, and the same things come up over and over again. The main thing in common between all of those articles is that they actually ignore the evidence that endogenous retroviruses originate from retroviruses in the environment.
No.
so
creationists ignoring things that disprove their argument. I don't know about that, Barry.
But
I'm incredulous.
It's just ignoring. They find ways to ignore the evidence and talk about other stuff. It's like distress flares, or it's just fire all the smoke screens.
Austin and Barry, I just wonder, as just a maybe a format for today, just to get your views on this. We get Barry to maybe just talk a little bit about what ERVs are.
Sure.
Just the basic stuff, just for the audience who kind of listens to this later. Just
Yep.
And then later, Barry, because I think between Austin, myself, and even yourself, Barry, we know what the arguments on the creation is. We know what they're going to say. So let's between all three of us, we'll throw out, we'll pretend to be creationists, and we'll throw out what the common arguments are. And then between all of us and Barry, what is the best way to rebut these arguments? For example, you mentioned the part about ERVs. They forget that they're actually parts or extinct might not be the right words, but fragments of extinct parts of viruses, right?
Yes.
I have heard an argument from, I think, Donnie from Standing for Truth that says, well, that makes sense, because they are, they fight off other ERVs. God created them that way, so they can use the mechanisms of the viruses to be part of the immune system. So I have heard explanations for this kind of stuff. So maybe give an introduction to what ERVs are. Between all three of us, we'll throw the common rebuttals back on what creationists will say, and we'll just slap them down. Does that kind of make sense?
Yeah, that sounds good. That sounds good.
Okay, great.
Yeah. Okay, let's start with what retrovirus actually means. Everyone, especially after COVID, knows what a virus is.
Yes.
But a retrovirus refers to what was called the common view, the word escapes me for the moment, that DNA produces RNA, produces amino acids. That's the dogma, the common dogma of biology. And it was a tongue-in-cheek name. The information always flows in that direction. Retroviruses are retro, they go in the opposite direction. And it was discovered, it became well known, retroviruses became known at the outbreak of the HIV pandemic, but they were known about before. It was identifying that HIV is actually a retrovirus, which made it come into public consciousness, what retroviruses do. What retroviruses do, and this is the essential part of it, is that they have their genomes, which are normally in the outside environment, are in the form of RNA. They invade a host cell, make the host cell convert their genome into a DNA version of the same genome, and then insert it into the host cell's own DNA. They do that in order to hijack the host cell and make it create more viruses by copying the viral DNA. So, and that is what we mean by endogenous. It's inside the genes. It's part of our genomes. And a common figure given is that about 8% of all our DNA is of viral origin, retroviral origin. So that's what retroviruses do. They get into your cell, they get into your DNA, insert their genome in, and make your cells produce more viruses. That's their replication cycle.
Yep.
Okay. So the thing about them is that they cannot insert in any predictable way, in any predictable location in your DNA. Nevertheless, when we sequenced our DNA, and they've done that with chimps and more creatures, they found that these bits of retroviral genetics are in identical places, going from cell to cell in an individual, comparing individual to individual within a species, and also going from cell to cell and comparing the DNA between different species. Notably, in the first case, it was between chimpanzees and humans, which were the first species to have their whole genomes sequenced. So once they were, aha, what have we got here? we've got viral DNA in exactly the same location in every cell. And given that that viral DNA is in fact viral, there's no other explanation than that they were put there by viruses. They're in the same places because they are inherited. We can still get viral DNA into our own DNA from the environment, but it goes in different places in our DNA, in different cells. Sometimes in one cell, sometimes not. A cell isn't infected, but these endogenous retroviruses are in the same locations in every single nuclear cell in our bodies. So that they came from inheritance is the only viable explanation for it. Okay.
Yep.
So
and Barry, if I can maybe just add to that. Sorry, Austin, I'll just
go ahead. Yeah.
If I can just clear that. So if we talk about, I think they say the human and chimpanzee split was 6 million years ago, right? If we go back, even past 6 million years ago, what you're saying is that there was a viral infection in the gamete cells. So yes, we see ERVs insert in a whole bunch of other cells, but unless it actually inserts in the gamete cells, so the sperm or the egg, it's not actually going to be, reproduced into the next generation. So there was an ERV event that occurred in the gamete cells, and that's why you see this replicated right up to us today. I mean, if it's not in the gamete cells, you're not going to see it. So it's even, whatever the percentage of ERV happened, it's even more than that in terms of the chances are less, because it has to be the gamete cells, right?
Yes.
And I think the other key point that kind of Barry brought up is we're not talking about 1 or 2 or even 10. We're talking about 8% of the human genome
that's right.
is full of ERVs that are in the exact same location, in the exact same chromosome that's across humans and chimps. And I don't know, and I'm just trying to remember the percentage of similarities between humans and chimpanzees, but we share something like over 99% of ERVs between human and chimps. And then you go back further. So if you think of this common descent evolutionary tree, you go back further from the chimpanzees to the gorillas, which I think they're saying 12, 13 million years ago was a diversion, right? We share something like 96%. So, and you go back to a mice, which is, I don't even know how long that is, but that's like 60% of change. So you're getting this really beautiful nested hierarchy of where an ERV
matches the traditional phylogenetics.
Exactly. And that's why we're saying that it's shared ancestry, because it's the best explanation, right? And so I just wanted to kind of
just to make one thing clear. Just to make sure we get one thing clear, we're not talking about one virus. We're talking about a whole bunch of them, different types of viruses. They would be named different diseases, and the numbers. So these come from multitude of ancestors, of common ancestors, our own ancestors, but it's not just a virus. Some creationists try to get away with that, saying, oh, you found a virus. We haven't found a virus. Some estimates say there are some 200,000 ERV and ERV fragments in our genomes. Some more recognizable than others, some more complete with others than others, which is what you expect from erosion, from mutation, and so forth. But the sheer number and the number that are not shared between chimps and humans is tiny compared to the number that are shared. It's like 6 to low 8. Hundreds compared to hundreds of thousands.
There's like 214 total endogenous retroviruses. There's 3 that only chimps have, and like 5 that only humans have, but the other 206 something are shared between us. It's like if you, like Ian said, if you went 6 million years back into Miocene apes, our ancestors got infected with viruses. And their offspring, their descendants, still have those viruses in matching places in the genome, even though there was a species split. So like chimps and bonobos and humans, they share over 200 of these, retroviral kind of scholars on the DNA. And
just want to
the odds
clarify.
of that happening by pure chance, like the mathematical odds of that are virtually,
I have
it's
a question for
asymptotically
Barry.
0%. It's like 1 over like 10 to the
ridiculous amount.
something power. It's an absurdly large denominator.
Yeah,
If
sorry,
I could
Ian,
I just want to ask Barry a question as well, because I had let's clarify the numbers here. I have in my head not 200. I have in my head that the number of ERVs shared between humans and chimps are numbering in the hundreds of thousands. Like we're talking about.
Yeah, I've seen a figure of 200,000.
Yeah, 200,000 was what I have in mind. Yeah, so I know you said like in the hundreds, Austin, but I mean, keep in mind we're talking about 8% of the human genome, so 200 just seems like a little bit less, right? So so gene fragments or ERVs that look like retrovirus, we're talking about hundreds of thousands here that are shared.
It's not always.
And your number, your probability number there becomes much worth. And you know how creationists love big numbers. Like they'll say things like, what is the probability that X number of chromosomes become exactly the way, proteins become exactly the way this? So what's the probability that life evolve? It's 1 times 10 to the power. Well, they love big numbers, right?
Yeah.
So the question then is, if you think this is a coincidence of hundreds of thousands of ERVs that's shared between humans and chimps, forget what's shared across the other animals. That's a massive number in terms of the coincidence that would actually happen. So, creationists love big numbers. There's a big number for you to try to explain.
We talked about going back in time and it matching the conventional evolutionary tree of relationships between creatures. And if we can use data from ERVs to look at erosion of ERV through mutation. So studying that can shed light on when speciations occurred, not just that they occurred, but at what point in time. We can estimate from the differences in the similar ERVs, this one compared to the other one is different. It's been eroded by mutation. That mutation occurs at a certain rate, and we can tell how long ago those two species diverged.
Okay.
It's very interesting, Gary, because I know that creationists say a lot, when did the ancestor become a human? Well, show me the exact one. Show me which. And it's not really exactly how evolution works. It's very gradual. It's not like at any point, like a Miocene ape just popped out a human. That's not how it works.
Well, yes, but that reflects on their conception of what a species is. It's like a fixed, immovable quality. In each individual species, because they're all created separately, they're all special creations in one way or another. They're all fundamentally different in one particular way. So it doesn't tie with their thinking that one species can merge into another almost imperceptibly and over a large population. Is just not the picture that they are equipped to take in. They still are looking for some magical ping that converts a pre-human to a human in one jump in one generation, let alone numerous generations in a huge population of creatures. So, which is the actual picture. They do not have the mental model, the construct, in order to, let alone agree to it, but just to comprehend it. Even if they disagree, they do not have the mental equipment to comprehend what evolution is saying.
Right. So Barry, let me throw in.
Hello, Mike.
Of the most. Hey, Mike, how you doing? Speed.
Okay, Mike.
Thank you.
Hey, I'm just here stalking Ian.
No, that's okay. That's okay. Hey, so Barry, I'm going to throw in the most common objection that I always hear from a creationist about ERVs, right? So we've talked a lot about what they are, so thank you for that, Barry. But also we talked about some of the probabilities of it, sharing, etc. One of the things they'll say is, well, evolutionists, this is where you're making the mistake that they're not functional. God created those ERVs exactly as they were. and humans and chimps had the same environment. They ate fruits or whatever it is, and that's why they all have the same ERV. So what would you say to that, Barry?
Okay, it's one of their main, most common attempts to object to the evidence, and particularly the intelligent design people cling on to this idea that there is no such thing as junk DNA. So it's all purposely designed, and if it's not functional, it's due to either the fall, if you're that way inclined, or to degeneration, negative entropy, genetic entropy, and all of that. Okay, first of all, nobody has ever said that the evidence for evolution from ERVs is dependent on all ERV genetics being non-functional. It's not the case. Nobody has ever said that. And besides, those people who have found function in parts of ERVs have not been non-evolutionists. They have not been creationists. The creationists haven't done any research on it. All they've done is to piggyback and exploit the findings of real scientists. So yes, there are functions that have been found in ERVs. They are always parts of ERVs. They are partly functional. Those ones that are, many are not. There's no evidence that the majority are functional in any major way. And the most common indication of functionality in ERV is genetic promoters. An ERV, a virus, a retrovirus that gets into your genome, needs to get the host to copy or to read its DNA in order to make the viral product. In order to make the cell read the viral DNA, it needs a promoter. It needs to be made to read, look here, go and read this piece of DNA and produce the RNA from it. And that's how normal cell functions operate.
Yeah.
A virus could not replicate without inserting promoters as well as the more conventional retroviral genes. So it's no real surprise that large numbers of promoters are of retroviral origin, and they actually have they do something. Whether it's a function or not, it's doing it.
Yeah, Barry, I would probably add to that. Also, I agree with what you're saying, but I want to talk about the numbers as well, right? So if you look through all the I think the ERVs were first discovered back in the late 60s, right? So we've got 60, 70 years of ERV research going on, and 200,000 ERVs, and all we've actually found are only a handful that actually have that actually do anything functional in the human. So the ones that the creationists always cite as example, they only cite 2 or 3. Keep in mind.
Yes.
If it's functional, as you say, from intelligent design crowd, or it was actually created by God, you'd expect more than a handful to actually do something. But we've only found a handful. The one that they always cite is the one that's actually common to the development of the placenta. The one that actually calls for cytosine 1. And you know what? As an evolutionist, that ERV was inserted obviously in the early days of mammalian
placental mammals.
You would expect with a fair chunk of a creature's genome to have ERVs, that the body would have had mutations that made one of them functional in some state. So it makes sense that out of all that, one happened to be functional, great. But the other handfuls are really, as Barry said, are only related to gene expression. So they're not you don't need it to be there. It obviously helps it to have to be there, but it's obviously mutations occurred, made it slightly beneficial in terms of function, and it became a gene expression. It didn't code for protein, but so it's only a handful is what I'm saying, Barry. You've got 200,000 odd that shift between humans and chimps, and we're only talking about a handful. Now, if creationists are correct and that they're all functional, well, show them to me. Show me more than 5.
That's right.
Show me 50. They can't do it, right? So and the other expression, and most of those gene expressions that we've seen that are functional, Barry, are usually related to the immune system, right?
Yes.
Now that makes sense. ERVs, retroviruses, what, as Barry kind of said earlier in this conversation, what do? They're there to insert into the cell, and basically the part of the virus would then fight off other viruses as well, because they want that cell to be replicate other cells as well. So the mechanisms that fight off other viruses, it makes sense that that's still there, and that actually is co-opted by the body to be part of the immune system of the body. Makes perfect sense to me, right? But at the end of the day, out of 200,000, we're only finding a handful of it.
Well, we may find more. You might find more and more subtle effects of, if it's an effect that could create, that's a function. It's an effect. And evolution will find ways round. We'll use things, and let's look at the syncytins. So I don't know if I'm pronouncing it right. The ones that form placenta. Placentae. So all placental mammals have these types of genes, not always the same ones. They have different ones. Some have several, which are not closely related to one another. Human beings have that. Genes that go into the making of placentae. But the thing is, well, several things. One, no complete ERV, complete functional ERV exists. If you had one in your sperm cell or your egg cell, you would never produce a viable organism from it. You wouldn't come to term. It wouldn't even develop, probably. So function is partial. Okay, if we look at one of the bona fide retroviral genes, the ENV gene, that is used, for example, by HIV. And what it does is a very clever trick. It absorbs T cells, which are trying to fight it. It absorbs them into one nucleated cell, just meaning a cell where the attacking cells have all been gobbled up and combined together. And it's the way HIV deals with attack from immune cells, the immune T cells. What a coincidence. You look at a syncytin gene to make a placenta. It's in exactly the same relative position in the genome, in the viral genome, as the ENV gene is. And it does a very, very similar thing. It absorbs other cells into a multi-nucleated cell, which makes the placenta. And it is such a coincidence. The other thing is that all syncytins, they're not all the same gene. In different placental lineages, you have different syncytins deriving from different retroviruses. So everything indicates that particular, particularly useful gene originated from a native ENV gene, environment gene, from a retrovirus. So the fact that it's functioned like any other function, it's been hijacked, been reused by evolution. Evolution has never preserved a fully functional retrovirus in our genomes. Otherwise, it wouldn't work.
That makes a lot of sense to me. As to how I understand it, if an endogenous retroviral line of genetic information were completely functional, basically the cell would still be producing the viral product, right? New viruses
out copies of viruses
still be infected.
Yeah. And they've done this. So they've done a fascinating experiment, which has been nicknamed the Phoenix virus. And this is really telling evidence. There are a bunch of ERVs which are relatively intact. They're not too eroded by mutation. That's why we think they're fairly recent in our genomes. But they're all very, very similar. And the idea is that they all have a common origin. They're all from the same viral infection, but we have multiple copies of it in our genomes. And each of them is non-functional because they've received mutations, other interference, in different ways. What they did is to string the genomes of each of these ERVs, one alongside another, and taken what's the most common base at this position in the genome, common to all of these ERVs. What's the next? Go along. What's the next common one? What's the next common one? And they produced what they called a consensus sequence, the sequence that the most numbers of ERVs agree upon. And the consensus sequence, the idea is that we've rubbed out all of the mutations, all of the faults, and so on, and we're getting back to what the original virus was. Guess what? They produced it. And they put it in a petri dish and fed it, and it acted like a bona fide retrovirus. It invaded cells, and those cells dutifully started churning out copies of the retrovirus. So they resurrected the original virus, which is why it's been dubbed the Phoenix virus. It's one of the most telling lines of evidence that shows that ERVs have a viral origin. And there are plenty of other indications of viral, why it's a viral origin, which I can go into.
And also this is a experiment from memory. I think there was the HERVK family of ERVs.
Yeah,
now the interesting thing about this is when they actually put this ERV together and replicated it, it acted exactly like what a cancer cell would do. You know, so and the interesting thing is these ERVs have actually been pointed to as one of the reasons why cancer spreads very quickly through the body. So, you know, the function that it was as a virus, even though it's a fragment of it, it was still kind of doing that in a human body. And in this example, it wasn't beneficial. Obviously, it had a negative impact on the human body.
That's right.
But it actually replicated like a cancer when we built it, and that's kind of what it did as in the body, in that it actually aided cancer cells and basically. You know, replicated within that.
That's right.
So it's a very interesting experiment.
So what you're telling me is scientists made a prediction, and then they constructed an experiment to test this prediction, and they found data that confirmed the prediction.
Exactly.
Gosh,
don't be a smart ass.
Classic good science.
About being the gold standard of science or something like that.
When creationists say that evolutionary science is not science, it's not based on real science, you cannot get a better example of real science.
Barry, it's just your religion, okay? It's just your religion. Hey, and stop being a smart ass, Austin. Stop being a smart ass. I can see your name. Hey, Barry, I'm going to throw you a second common objection from basically based on what you said, right? So this is some creationists. So we talked about creationists saying, oh, you know, it's all functional. God created this way. So let's throw in an objection, a common objection to what you've just said, Barry, right? Which is, well, God created that way when it was all perfect. So back in the days of Eden, animals and whatever, because chimpanzees and humans are exactly the same. So God created all these ERVs perfect. They were functional back then because they had an important element because everyone was perfect. Then Adam sinned, Eve sinned, and ever since then, we've had evil come into the world, death come into the world, and genetic entropy happen. And that's why most of them are now non-functional because of Adam's sin. So what would you say to that as an explanation as to why it's non-functional now?
It's ironic, really, because scientists are criticized for not doing real science. And where's your evidence? Where are the facts? What's the outcome? Have you made a prediction? Have you tested it? Has it tested out right? Is that being applied to the fall? So there's something a little amiss. Somebody who says, oh, well, it could have been this. It could have been that, you know, that everything was created perfect and it's all gone wrong. Where is your science? Where is your evidence for what you propose? There is none whatsoever. So it's rather ironic and hypocritical to criticize science when you have no science whatsoever behind what you are thinking. Besides that, I mentioned the Phoenix virus case, but there are a lot of other indications for the idea that ERVs are of retroviral origin. And some of it gets a little bit technical, but not too much, I don't think. There is something called codon bias. When you have three base pairs, CAGT, you have three of those in a row, that codes for an amino acid. And that bunch of three is like a little word, a three-letter word. Each of those three-letter words could be different out of C, G, A, and T, whatever combination you like. Each one of those, or nearly all of those, will code for an amino acid that goes on to make a protein and blah, blah, blah. It turns out that there are several ways of coding for a particular amino acid. So those three-letter words might be CGA, or it may be CTA, for example. I'm probably wrong, but you get the idea. There are different versions or accents in the codons being used to specify amino acids. And it turns out that mammalian DNA has one style of codons, uses one language, one accent. Viruses use another one, a different one. And it's called codon bias. There is a signature in the codons themselves that this is likely to have come from native mammalian DNA. And this one is more likely to have come from viral DNA. So codon bias, it's something you can look up and learn. Anybody watching this can look it up and confirm what I say and get a better explanation than I can give here. But it is one other line of evidence that ERVs derive from retroviruses. Another one is the koala retrovirus. It's been nicknamed KORV. And this virus is actually in the process of endogenizing in koalas, which means that there are a large number of koalas without these ERVs. There are a large number of koalas with ERVs, but in different locations. And the explanation for that is that this is an ongoing endogenization. There are koalas without these ERVs, so they are not inherently a part of the original design of the koala genome. They're obviously come from outside. And there are various aspects to that. The explanation of what we know about KORV fits the endogenization scenario rather than the created one.
So you're telling me we can see the end result in comparative genomics.
Yes.
We can replicate the disease itself using this Phoenix experiment.
Yes.
We can watch the process happening
in real time.
Koala population. Wow, isn't God's design amazing, Barry?
Hey, so Austin and Barry, incredible. I like to keep things simple for our creationist colleagues because let's be honest, most of them, actually all of them, none of them that I have spoken to on YouTube have any formal education in any of the relevant science.
Yes.
Right? No one of them study that. So I like to always keep things simple to them.
Yeah, I do my best, but you inevitably get into some of the nitty-gritty details.
8% of the human genome.
The added problem is that they're like this. When you try to explain,
they don't know and they don't want to know, right? So they don't know, they don't want to know. But look, so my answer to this is just to keep it really simple. We've got 8% of the genome that are ERV shared between human and chimpanzees. If you assume they're all functional and they've all degraded, then please, Mr. Creationist, explain to me how such a huge percentage of your genome shared between chimpanzees have degraded in exactly the same way. So we're not talking about, 2 or 3 and, out of 5 that happen to degrade. We're talking about almost 200,000 of it. Has no function now. They appeared in the same spot. What are the probabilities that was all functional and that just happened to be those exact same ones across humans and chimpanzees are now no longer functional?
Yes.
So
why would a creator? Why would a creator create all of these ERVs and then let them become disused? Surely, originally they had a function, and how to let it all disappear? without
to your
mass
point, Barry,
extinction, etc.
that
Yep.
that is the 200,000 ERVs that are now exactly are no longer functional across humans and chimpanzees.
As
But
retroviruses.
let's
Yeah.
not remember, I think it's 600 or 700. Let's not forget about the HERVs. So that the ERVs that are only found in humans. So obviously evolution still happens, as we had that split between humans and Homo sapiens and chimpanzees. There were still ERVs that are coming into the gamete cells of humans. So there are
That's
still
right. It's going
ERVs
on all
that
the time.
don't appear in chimpanzees. So explain, I think 600 or 700 of them, explain that small percentage ERVs that are also not functional, but also don't appear in chimpanzees. So we've got the vast bulk of it that are shared, that are, have degraded in obviously the same way. But we've got a small percentage that is only found in humans, also degraded as well. So Mr. Creation, explain that.
Yes, that's right. That's right.
Yeah.
I mean, we can explain. I've got on my blog a list of questions that a design hypothesis would raise. For example, why is there a viral codon bias is one of the questions. What are these viral enzymes actually for? What is integrase for? We know what it's for. It's for splitting open the DNA, inserting the retroviral DNA and closing it all up again so that the host comes along and copies it. Why? What other reason could there possibly be for that integrase enzyme? Reverse transcriptase is one that converts the retroviral RNA genome into DNA before it gets inserted into the cells. What is that designed for? There are some, 15 questions I've written out on a page that a design hypothesis has to be able to answer to be a viable hypothesis. Many creationists have attempted to answer one or two of those questions. Mostly they just bypass them. They don't even try. The ones that do make a brave effort, but their objections and their comments just make no sense. And I've got some examples of their attempts on the blog as well. So there's a list of questions. Yes. If it's designed, why this? Why that? What's that for? They can't answer.
And Barry, I
put my answers, which are all easily answered by what I call the endogenization hypothesis.
And Barry, your what you've raised there is I have used as the absolute #1 best evidence for common ancestry. We're all talking about ERVs now. We're focused like laser-like on 8% of the genome, right? Yeah. But let's pull it out. And if you look at more than just ERVs, so Barry alluded to this, you've got sign and line elements, you've got ALU elements, you've got pseudogenes. So exactly the same arguments and discussions we're doing in ERVs. It's the same thing. And then you're not talking about 8% of human genome now. You're talking something like 66, 67%. So the vast majority of the human genome shows shared elements between human and chimpanzee. So it's not just ERVs, ALU elements, all right? I think lines, it's like 1 million lines. And to what to what Barry's saying, these are, ALUs, for example, are jumping genes. They're genes that kind of jump around the body as well. And when they jump, they leave a very small fragment of itself in the original location. They're so small, it absolutely does nothing at all. So we know that does nothing at all. Right? And we can track not just where the ALUs are between human and chimpanzees, but where they used to be, by these small small fragments of its DNA. And we can see the shared between humans and chimpanzees, not just the ALUs, but where they've also jumped from as well. And we're talking about, I think ALUs are something like almost, from memory, I don't know, what is it, 20%? 20% of the are ALUs, right? So explain that. So we're talking about 8%. The same argument can be used to expand it to almost 70% of the human genome. So I think you've got that here, are you? What have you got there?
I can't begin to say how one of the more science degree holding creationists might try to argue with this, but I can tell you, I can make a guess of how the layman creationist might try to argue with this. And they'll just say that stuff was just put there by Satan to test your faith.
Yes.
And you have to reject it and keep believing in Jesus. And if you don't, you're going to burn in hell.
Well, that's
Right.
like people saying dinosaur.
The smarter creationists, like intelligent design proponents, they would say that actually all of this is functional, so it's not accidental. That's what they would say.
Well, that's
And
demonstrably false.
And to your point, also the dumb ones, the ones that are not educated, will just resort to magic. In which case, it's not a discussion. We're not talking science anymore, so I'm not gonna talk about magic. If you're gonna rely on magic, anything's possible, right?
You're right.
If you're gonna talk about science,
I just like to throw in one thing that the discussion has reminded me of. It's a guy, Graham Finlay, if you don't know him, is New Zealander. He is a world expert on virology and genetics, and he wrote a book that talks about ERVs, explains everything about ERVs, but also ALUs, lines, signs, pseudogenes, and his book is called Human Evolution, Graham Finlay, and I recommend that to anybody.
Yes,
Graham
Finlay?
a scientist.
Is that with an F?
He happens to be a lay preacher, a Christian lay preacher. You'll find him on BioLogos, and you'll find him in universities, well-regarded expert in these matters, and he's a Christian. So this idea that evolutionists are atheistic and they are anti-God and all of this, no, we're interested in the truth. What is real, what is true. As I would recommend that book to anybody, the Graham Finlay, and make sure there's a note of that at the foot of the video when it goes out.
Will do.
It goes into things in a lot of more detail than my blog or this discussion could.
I'm going to make a small correction. I did say the ALUs were, I think from memory, I said it was 20%, but just based on the slide that Speed just put up there, ALUs are a component of SINEs, short interspersed nucleic. I can't remember what these stands for, right?
Yeah.
Obviously, that makes up 13%, so it's something less than 13% because ALUs are the majority of SINEs. So it's something up to 13%. I don't know what the exact number is, but it wasn't 20. So just a small correction.
I can tell you.
And pseudo.
Mike, you've been waiting very patiently and sharing the graphs with us.
Yes, indeed.
Very useful. So I'm gonna I throw in when you're ready, guys. I'll throw in another example, another argument that the creationists would kind of say, right? If you like. Okay, so we talked about the ones that says no, they're all functional. And then we talked about okay, they used to be all functional, but they've degraded. All right. So I know Donnie likes this one because he's probably he's smarter than the average questioner. So he tries to back it up with some science, and he wrote a whole book about this as well. Which
I've reviewed his books, a couple of them. Yes,
well, I love that, Barry.
I've actually got it here, to be honest. So I've read it. So that's why.
How would you rate his books, Barry? Would you give them a high rating, or would you say it needs
I'd put an Amazon rating up there, the lowest possible one. It's the usual crud. It really is. And he's not a very pleasant chap. He's gone behind my back and attacked me without letting me know, not letting me to come back to him, blocked me from all his outlets, so I can't respond to him. And there are a number of pages on my blog. What's his name? Donnie. Donnie Deals. And what's his Matt? And I call them the Beavis and Butthead of creationism.
It's raw Matt.
This book actually, at least it explains the creationist thinking. Okay, so and I got it from this book as well. One of the, I guess what I would say, the third most common argument about ERVs, right? So what Donnie argues is a little bit more nuanced than just it was all created perfectly and it's all degraded because of genetic entropy or whatever. What he'll say is that there are hot spots in the human genome that are
that's a common one as well.
That are likely to have mutations, and because of these hot spots, that's where you see the ERVs kind of attach itself to, etc. And that's why you're getting that commonness that's occurring between human and chimpanzees. So as I said, we might not agree with the science on this book, but what it does highlight, at least to me in one place, is the creationist arguments around ERVs.
Objections they have.
If I kind of said to you, Barry, what is your rebuttal against what Donnie's saying around mutational hotspots? And that's where the ERVs are.
Okay. Talking about integration site specificity. There are a number of studies that have been done, especially when HIV came to light, where people were furiously studying integrase and where retroviruses integrate with the host DNA. So they did surveys of integrations of viruses into particular cells. They used the same cell type in the same individuals, exactly the same genetics and so on. And they found typically something like a repetition of integration site one time out of every 500. So the chances of two cells getting an integration to that particular DNA locus, that individual locus. And by the way, when we talk about a locus, we're talking about resolution down to single base pair. So little regions don't matter. We're not talking about little regions. We're talking about specific loci, specific locations within the DNA. That will only occur something like 1 in so many hundred, typically 1 in 400, 500. To get 2, by coincidence, 1 in 1 over 400 times 400. To get 3, 1 over 400 times 400 times 400. To get 1,000, 1 over 400 to the power of, you can see that the numbers do not work. There are preferred areas where certain viruses will tend to integrate in certain types of regions, near a coding gene, near an empty space in the DNA, which according to the DI doesn't exist, but nevertheless, different viruses will tend to will integrate in, careful with my words, they will tend to integrate in certain types of regions. But again, we're talking about types of regions rather than specific loci. Right. I use an analogy with reports of road traffic accidents. And you think of a retrovirus integrating into the DNA is equivalent to a road traffic accident. Okay, you're looking at a whole load of road traffic accidents. And you say, in an accident hotspot, for example, just like there's an integration hotspot in the DNA. Are those accidents different accidents, or are they reports of the same accident, i.e. copies of the same accident report? Let's have a look at the details of them. Is it in exactly the same place or just in the same general area, one end of the street or the other end of the street? Did it involve the same vehicles? Did it involve the same occupants? Only if all of those are the same, you can conclude that that is a copy of an accident report you've seen before. Only when all of those details tally do you know it's the same. In the case of integrations from retroviruses, if they are in the same locus and they're the same DNA insert, they are copies of the same endogenization event. Endogenization event meaning the first integration into a cell, which is then subsequently copied from cell to cell and generation to generation. So they're copies, rather like the accident reports being identical are copies of one another. So that is a common objection. It's not, we get ARVs in the same location. No, they're in similar locations. No, we're not talking about similar locations. We're talking about precisely down to base pair resolution, exactly the same locations. And only inheritance can produce that effect.
That's one thing, yeah, that they kind of try to imply that other processes besides inheritance can produce these things, like, oh, similar lifestyle. Like, I don't see how that can give two descendants of the same population, that same insertion.
Yes. Yeah.
Having a similar diet isn't going to affect your genome, as far as I understand.
This goes back to the surveys. I mentioned the surveys of integration sites, because people were desperately trying to weigh in what are the regularities surrounding this retrovirus phenomenon. We can gain any information that may arm us to combat these viruses. So one of them was, where do they like to integrate? Can we concentrate our drugs in trying to prevent integration in those areas? So they did very, very careful surveys of integration sites. And what they found was that the separate integrations, rather than from outside for external exogenous retroviruses, viruses coming from the environment and infecting you, even when you're talking about an organ with exactly the same cell, exactly the same DNA in every cell, the retroviruses integrate in different locations within that individual, within that organ, within that tissue type. They could not find repetition of integration sites, not enough to rescue that line of argument from creationists that retroviruses will tend to integrate in particular specific locations. They don't.
And you know what else I like about that? That indicates a degree of randomness, correct?
Yes.
That there is, they can randomly insert at different locations. Yes. And that is a powerful argument against design, because how can you have a design that is random? Randomness is the opposite of design. It's only possible to consider randomness design by question begging. As far as I can tell, there's no logical way to say that a random event was designed.
That's right. And that's one of the... Go ahead.
I want to point out that Matt and I think Donnie and John Maddox have all claimed that these retroviruses are, that the way they work is that when you're walking around, doing stuff and eating carrots, they will jump into a certain location and do some function, okay? Which they don't, because they jump, even the hot spots. It's like you said,
it's a statistical tendency to go in a certain areas. It's not specific locations that it goes into. Yeah. Sorry, Mike, you've frozen. I don't know if you can hear me. You've frozen on screen.
He's telling us about the fish he caught. Like, it was this big.
Yes. Maybe he'll come back, but I can't see or hear him at the moment. He's frozen. Yeah. There we are. Okay, so you're back. Hi. I'm trying to think of other common objections, and I think I've dealt with them all in my writings, but it's hard to just come up with them all off the cuff. But
there's the idea that it was designed for Adam, that they designed it right. Well, why would you design something that jumped around in the genome? Particularly jumped around randomly, and that doesn't make any sense. And then after the fall, of course, they jumped around less precisely and started duplicating. And why 500,000 lines and 1 million signs? I mean, why do we need that?
Yes.
I wanted to tell one story before. Barry, you're wonderful. Your blog is wonderful, and I did not know about you and just found out about you. But just for the hell of it, one time, I don't know if you're familiar with NCBI. I can show it here. Sure. NCBI is a site where we have the human genome, right? And you can't see that too well, but what I'm showing here is a gene. And if you can see those darker green blobs right there, Those are all exons, and in between are called introns, the part that gets spliced out. So it's a part that's mostly junk DNA. I know people don't like that term, but I do. And so I took a random gene and I went through and I did a cross. I used a chimpanzee and a human being, and I used a mouse as my outlier, right, as my third one. And I went, and now on NCBI, and I don't know how to turn it on again because they changed their damn interface again, but you can turn on a little thing that will show you where all the signs and lines are in the human genome. And it looks like a big mess of, green blobs, right? Because they're all over. And one of the places you'll find them is after, like I found one after the first exon in the first intron, right? Which is the piece that normally gets clipped out and thrown away. So I took that particular line and I went and I did a comparison between humans and chimps. And I found like 1 or 2 nucleotides difference, but it was still there. And then I went and looked at the mouse, and lo and behold, it wasn't in the mouse. It had been inserted somewhere along the lineage when we split from a common ancestor with a mouse. And I just randomly picked one of these 500,000 lines in the human genome. And I'm guessing if you picked any one of them, you'd end up with roughly the same results.
Right.
And if you did about 100 of them, you'd have an incredible little bit of information about phylogeny and how the tree works. So that is something that, and I was amazed because I thought this ain't going to work. There's going to be no homology at all here. And it was just textbook, textbook.
Yes,
evolution.
yes. And it's a general point. All of these studies and all the detailed work that goes into all of this sheds light on our evolutionary history. So it's very, very interesting in that sense. But comparing to creationism, who will just wave their hands around and say, God did this. Why did God do that? Why did it do it this way? Why put all of this detail, which is consistent with the scientific version of events? Why put it and why mimic the evolutionary version unnecessarily? They do not address it. It's all, the devil is in the details, and that is why they don't look at the details, because all the details confirm the evolutionary story. That's just a general point.
It's such good evidence for evolution. It makes so much sense through the lens of evolution that I have a hard time even thinking about how a creationist would try to
refuse it. They just,
which is why they mostly just ignore it and avoid it.
Yeah. It's hard to get into their way of thinking, and I've been at this game for a very long time, and I'm still rather baffled by how they can withstand this absolute avalanche of information, finely detailed information, which is all consistent with evolution.
I like that it gives us a way to test our ideas about how fossil species, extinct ones, how extant species relate to each other in a different line of inquiry. It's less. It's more than just comparing homology. We say, okay, these homologies look similar. Maybe these things have common ancestors, and then we go in and we look at the genomes of extant species, and we see these similarities, and if they match, the nested hierarchies in taxonomic phylogeny, on a molecular level, and that's very compelling. I can't even understand how a creationist can look at that and not see it through the lens.
Another general point is that creationists say, well, your theories are not being tested. They're being tested all the time. As soon as we come upon an anomaly, somebody gets a Nobel Prize, or nobody has managed to disprove it. It's always possible, in theory, that something will come along, will throw a spanner in the works, and it doesn't fit. It doesn't happen. It's, we've been at this game for since 1859, since Origin of Species, and nothing has come up which gainsays the evolutionary story. It's been modified, it's been expanded. Some things we've got wrong, we've corrected, but nothing has overturned the evolutionary view of how life evolves.
Yeah.
Anyway, guys, I don't know how long we're going to go on.
I think we're signaling the end here.
I think we are coming
if I can ask one thing, Barry.
Yeah.
What do you think the common person? What's like if you could impart one point about ERVs to the common person to absolutely remember? Yes. What would that be?
Oh, that's a difficult question, really.
Mutations.
I think in order to gain an understanding requires a little bit of effort on your part to understand what we're talking about, but not an enormous amount. It's not rocket science to understand how a virus can enter your cell and insert itself into your genome. It's not rocket science to understand that this viral genetics is the same in every case because of heredity. That's the way heredity works. So they need to be able to study how retroviruses work, what they do, just at a basic level, and they need to understand how and why heredity will put the same genetics in every cell of your body, and they're in the same location in every cell of your conspecifics, and also in most cases in your closely related species. So it takes a little effort to be able to appreciate that, but not an enormous one. If you have an open enough mind just to look at it, and really, if you're creationists, you ought to look at it. So even if nothing else, just to equip yourself with possible objections to it. You need to have a certain basic understanding of what we're on about in order to try and criticize it. You can't just say, oh, no, God did it. That's not
common design. Common design will do that too, Barry.
And design, or God did it.
Yeah, with that example that I did, where I took the line, the line 1, and I found that sequence, right? And I found it in chimpanzees and humans. Now, the way to test that, so the creationist comeback would be that, that little piece of intron had to be functional. It has to do something, right? That's why it's in the chimp. It makes us have like chimp-like fingers or whatever. So what you do is you look across humans, right? At that same region, and what you're going to find is you're going to find a lot of mutational variation, a little bit, because of course, it takes a long time to hit that one spot, but you can find mutational variation. And the other thing is you can test it to see if that little piece of RNA it ever gets out into the cell, or gets out of the nucleus and does anything. And there's all these ways to test for function. And somebody was talking about how many of these things we found to be functional. I think I had a table one time of about 28 of them. 28 out of several million of these jumping genes in our genome. I mean, if the creationists want to dispel evolution, they have to show high functionality. They're going to have to take like 10, 20% of those things and tell us why they're functional. And you do that by telling how fast things mutate. So if things mutate between you and I, then obviously that piece is not functional,
Yes.
And
it's not conserved regions. Yeah. Right. Listen, guys, and I mean, I think I'll have to call a halt to it for myself for the moment. You can continue if you want to. It's been really enjoyable seeing you, and I hope we do it again very soon. But I think we've covered the ERV part pretty well, and I'll get together with you, Austin, about putting some notes together on any video that comes out, and how we share out and publicize our discussion. So thank you very much, guys.
Great talking to you, Barry.
I really hope we'll continue the discussion and find other things to talk about at a future occasion shortly.
Thanks. It's good to meet you too, Barry.
Yeah, good to meet you all. Thank you very much, guys. Okay, then. Bye for now. Bye-bye.
Bye. Actually, there's one thing interesting about ERVs that we didn't really get to, and that is, and Barry kind of brought it up, ERVs were obviously viruses. One of the things that viruses would do when they invaded your cell was they would put out proteins to stop other viruses from getting to that cell, and then it would then replicate itself, right? And that's why, we see a lot of ERVs that have been co-opted by the body. When I say a lot, we'll talk about, a handful that have been co-opted to for our body to do gene expressions that help with the immune system. I know some of the latest antibodies or anti-vaccines, those antiviral drugs that they've got against HIV. A lot of that came from some studies done on the HERV viruses. Basically, they've identified a particular gene family in the human endogenous retrovirus, HERVK, I think it is, where they've seen that those HERVK class ERVs actually produces proteins that actually interfered with the replication of the HIV virus. So they kind of developed that and produced these antivirals that we kind of use today that are highly effective in basically stopping HIV from spreading in the body. Now, I think that's a really interesting bit of research that we can probably look at in the future. So we know what ERVs do. Yes. We know that they have something to do with the immune system. The whole point about viruses in the past was to stop other viruses from invading that cell, so that cell can replicate its own virus. It's a really interesting piece of research to kind of use the tools of ERVs to stop basically other viruses from attacking the human body. So that's so I think that's an interesting thing that we can talk about.
And we have a system we have a system, this crab complex. It's a zinc finger complex that basically silences these things too. That works on silencing lines. I think lines and maybe signs. But there's always, so we've developed this immune system inside of us to silence these jumping genes, because of course they can be a real problem. And absolutely. There's something that you have to really think carefully about when you do this. One is that you've got the germ line, right, which is the first few divisions of your cell, that end up in sperm and in eggs. And then you have the soma line. So all of our lives, we've got this degradation of our genome by lines and signs. And of course, we have all these tools to repair the damage. But so it ends up in getting into the evolutionary germline much more rarely than it does in our soma line, which is what we, I'm 74 years into it, right? So I got a lot of jumping genes running around. But so that's important.
But all those jumps and breakages and repairs, they leave behind evidence, and studying that evidence, it's kind of like putting together clues at a crime scene. And ERVs are kind of like if you find a bullet that has congruent scratches at two different crime scenes, those bullets were both fired by the same firearm, because only the same firearm would leave those congruent scratches. It comes from the rifle, the barrel of the firearm. Sorry, not the rifle. Rifle's a different kind of firearm. The barrel, it's a part of the firearm that it scratches the bullet as it exits. And you can think of ERVs as being kind of similar, like a congruent scratch that was left behind by a viral infection on one of our ancestors. And because it exists in two different species, those species shared common ancestors. It's a really good line of evidence. It's phenomenal. And I think Ian, the idea that parts of ERVs have become useful in our genome as like viral prevention, that's a good that's a good refuter for a couple of other common creationist arguments, like, mutations happen, but they're only ever bad. They only ever harm, which is not true. They're contextual. So, a good example is sickle cell anemia. Yeah, it's a disease, and it hurts the person who has it, but it also makes them less likely to get malaria. So, in a way, contextually, it is something of a positive mutation.
So 2 points on that, Austin. The first point around creationists is you're going to have to get them to accept evolution first, because they'll just say God created it that way, and therefore, it doesn't have to evolve any function or whatever. God created that functional. The second thing about the sickle cell one is that it's all about probabilities, right? So it's only bad if both your chromosomes have it. Okay, so if one actually has the sickle cell and the other one doesn't, then it gives you immunity. Plus, you don't get the disease. It's only when you both actually have it. So if the mother and the father have it, and it comes together, the child will have it, the mutation, and they'll get that deformity in the red blood cells, which is actually bad. But if only the mother has it or the father has it, it comes together, you'll get the immunity of it, but you won't get the disease. So it's all about probability. So obviously, the offspring that has the both will be weeded out, but the majority of it that don't have it will still live on to have more offspring. So it's all about probabilities.
Yeah, it's really interesting
that this was a great discussion. I do believe this is some phenomenal slam dunk evidence for evolution. You almost just have to la to try to argue against it. I think this would be point number 58 on my list.
Yeah, before we go, I want to point out that paper that I sent you the name of. That's a really important paper. It's 10 things you need to know about endogenous retroviruses.
I'll add that to the link in the description after.
Yeah, this is an illustration that is the most wonderful little chart that tells you where all these viruses, how they're classified. Like here are the HERVs and the ERVs and the long terminal repeats and all of that. But there's so much to say about this subject, because these long terminal repeat viruses, they're also very, very important in how genomes get rewired and reshuffled, and why mice have different numbers of chromosomes than we do, because these things they affect their own kind of mutational evolution, right? And it's really interesting. I mean, this subject is so deep, you could spend your entire life on one of these branches studying them, and people do. So there's just so, and this is like, I think this is probably the most powerful proof of evolution, is looking at these things. And like I said, I just stuck a pin in a pile of 1.5 million viruses, and I found this perfect homology.
Yeah,
I mean, it matched the evolutionary story perfect.
Yes.
And you can stick a pin in any one of those damn things. That's pretty cool.
And Speed, this is where I sometimes get a little bit frustrated with the creationists, is that we talk about ERVs and they only focus on ERVs, and I said
not the rest of those.
They forget about also you got LTRs once again shared in exactly the same place, besides exactly the same ERVs. Across humans and chimpanzees and going forward, and then you build a nested hierarchy that way. But pick any category in the genome, and you can build that nested hierarchy exactly the same. I mean, what I was talking about in terms of ALUs, which is part of the SINES, when they jump, they leave behind something called an ALU remnant. So we know where it's jumped from and where it's jumped to, and and obviously where it's jumped from. Those ALU remnants, they do nothing. They're just like small fragments of shit. Just gets left behind. So we know they are not functional at all. All right. So we see these ALU remnants and we see the ALU elements in exactly the same place. And you've got to ask the question, is stop focusing on the ERVs. Whatever argument that we've given you in ERVs, expand that to everything else. So something like 70% of the human genome.
Yes.
Explain all of that, because now you've got a big number. Now you can't talk about 1 or 2 or 3 being functional. You've got to explain 70% of that. So that's where I think we get them. It's not just the ERVs.
And I get why they do that. The ERVs alone, that's like a knockout punch. Yeah. But if they can focus on that one knockout punch, they can maybe kind of stay on their feet. But when you expand it out to the ALUs and everything else, that's a barrage of knockout punches.
So this is where look at this. Pete and I are talking to someone who's, a former MMA fighter. So obviously your examples about knockout punch and that kind of stuff. So I'll use the example. ERVs are the knockout punch. Once the guy's knocked out, you're basically just, I don't even know if this is legal or something, but you're basically knocking them out even after they've been knocked out, you're killing them.
The referees should throw themselves between you and them by that point.
This little pie chart is really important. Lines make up 20% of our genome and signs 13%. I mean, we are filthy with these things, and it's really a rich field of study. So it's pretty cool. Anyway.
Indeed. So, Ian, our friend Maya really wants you to come on her channel and geek out with her some Sunday. So, I'm gonna have to get you in touch with her somehow. Do you have Discord, Ian?
No, I've last time we had one of these, I was properly criticized by Maya by not being a subscriber, and I have since rectified that.
Oh, good.
So Maya, on that, you should know I'm now a subscriber, and I have seen a couple of the shows and stuff. I do want to make a correction that I don't consider myself a geek. I'm more of a nerd. If you can see my book collection, it's all fantasy and science fiction. It's there's nothing tech related here at all. So yes, happy to, Maya. I will just work out when. Sunday for you, is it Sunday night? That's probably what time is it for you, Austin? Now is it
right now? It's about 1:30 in the afternoon where I am.
Right now it's 5:30 a.m. for me. So we started at 4 a.m. for me here. So
yeah.
if it's Sunday afternoon or thing, it's probably going to work out to be Monday late morning or whatever. So that works for me as well. And as you can see, I'm very happy to come in early in the morning my time, so don't worry about the flat earth. But yeah, so I'm happy to, and we'll work out when, and
I'll put you two in touch and we'll get that set up.
Yep. Thanks, are from the future. You are from the future.
I can tell you exactly what's going to happen after this.
Is he still president where you are in the future? Is he?
I'm not that far in the future, but so the tariffs are going to happen. He's going to backtrack on it. It's going to happen 3 or 4 more times. Don't worry.
Yeah.
It's going to happen. Yeah. The next day he's going to cancel it. It's going to, typical.
Oh yeah.
Yeah.
Yeah, we're riding the roller coaster, right?
Yeah.
Okay, guys.
Well, thanks so much for stopping by, Mike. Thanks to Barry for stopping by. Thanks to everybody in the audience for listening in as we geeked out about genetics for a little while. ERVs are phenomenal evidence for evolution. If a creationist tries to tell you evolution doesn't have any evidence or it's not testable, just throw the ERVs right in their face. And if you need to, tag me or Ian, and we'll come in and set them straight.
Yeah, but don't do what the chimpanzees do. It's not ERVs they're throwing to your face. It's one of these.
Yeah. All right. Thanks so much, fellas. I think we're going to end it there.
Okay.
See you often. Thanks
Everybody
again.
have a great day.
Bye.
See you guys. Bye.